Hi everyone. How are you doing? I hope everyone is doing good. So, welcome to another video of inheritance and variations. In this video, we are going to see another examples of variations from Mendelian genetics. So, the first example is the earlier two examples were complete dominance and incomplete dominance, which was discussed in the earlier video. Now, in today's video, we are going to see the remaining two examples, that is pleiotropy and multiple allelism. Okay, so let's begin straight away. What is pleiotropy? First, we can understand pleiotropy. It's the simple definition. So as you can first we'll see the representation which is there on the screen. You see that there is marked over here something as single gene. Okay. What is the effect of the single gene? It is having multiple traits. Okay. This gene is controlling or it's having an effect on three different characteristics. Trait 1, trait 2, trait 3, that is ABC being that. So basically, pleiotropy is one gene, but more than one function or more than one effect. Okay. Up till now, we were seeing, for example, one capital T. One capital T, for example, this is the allele for giving tallness. Okay. If I write one small t, this is the allele which is giving dwarfness. Okay, which will cause if the recessive the parent is homozygous recessive, then the plant will be dwarfness. So this particular gene is responsible for only giving dwarfness to the plant. So one gene, one trait. Okay, but pleiotropy is something different, which is not following Mendel's law. It is showing multiple effects or multiple functions. Gene is one, but it can have multiple effects. Let's see, trait 1, 2, 3, ABC. Okay, so this refers to an allele which is having more than one effect on the phenotype. You need to remember this. More than one effect on the phenotype. Okay, just now I explained with the example. So single gene exhibits multiple phenotypic expressions. Up till now, as regards to Mendelian genetics, one capital T, let's say, and a hybrid condition or a homozygous condition. This is responsible for only giving height to the plant. This is responsible for giving only height to the plant, that is tallness or dwarfness. But here the gene, the single gene will give multiple phenotypes. So how, what example that we will see in a short while when we do the example. So pleiotropy refers to the gene giving more than one effect or showing multiple phenotype. Okay. So one gene affects multiple characters. Obviously, it becomes difficult to predict because only one gene is involved. So we don't know that this trait is coming from which gene, or are these traits, linked to the expression of a single gene. So phenotypic effects are pleiotropic effects are difficult to predict because a gene that affects one trait often performs other also. So unknown functions cannot. It becomes very difficult to know the number of effects that they can have. But obviously, the science and research has advanced to such a level. There are certain examples. There are research being done wherein we know that this gene can affect multiple phenotypes. It has an effect on multiple characteristics. So that example, which is there in your syllabus, that you will see. So examples, for example, starch synthesis in pea, then phenylketonuria, sickle cell anemia. Okay, there should be from over. Sorry. Cystic cystic fibrosis. So these are some of the conditions. You can say some are diseases, some are just metabolic pathways, which show one gene is responsible for multiple characters. Okay. For example, if you will take a sickle cell anemia, which you are going to study in detail over here, because this is what is there in your syllabus as an example, so should be studied. We will be studying under pleiotropy as more in detail. So this sickle cell anemia can have an effect on multiple organs, as you can see. It can result into blindness, it can result into failure of the liver, or it can result into heart attack. So you see multiple phenotypes, multiple traits can be expressed because of the mutation of this single gene or some defect in this single gene. Okay. So one gene is responsible for giving you different characteristics. Though it's a disorder, but then that is what is you're talking all about. So example here, rapid breakdown of RBC causes anemia, leading to increased lethargy. Okay, one of the effect of the pleiotropic gene under the example of sickle cell anemia can be this rapid breakdown of the RBCs, which will result in anemia. Okay, there is loss of RBCs, and because of RBC low number of RBC, the amount of oxygen which needs to be transported to different parts of the body reduces, and that is why the person feels less energetic. He or she feels lethargic. So paralysis due to clotting of sickle cells in the vessels. We will see what happens actually in the sickle cells. These are the symptoms. That is the effect that we are getting introduced to. So what we know now that this pleiotropy is having multiple effect, multiple characteristics. So one characteristic, one effect is this, resulting into anemia. Another effect is paralysis or stroke can happen because of the irregular shape of the RBCs. Organs like liver, kidney, heart can be damaged due to clotting of the blood, accumulation of the blood cell. This is another effect of the gene which can take place. So these are some of the effect that has been put over here just to get yourself introduced. That what do we mean by having multiple effect? Okay, so if there is here with respect to mutation in this gene, then this gene, if it is not working properly, it can have an effect on different organs. It can bring about these symptoms. Okay, so let's begin. Let's understand the first example, that is sickle cell anemia. Okay, fine. First, let's understand what happens in sickle cell anemia. So in sickle cell anemia, there is irregular red blood cell, okay, caused by an abnormal hemoglobin, irregular red blood cells. Usually the red blood cells are circular in nature, as you can see in the diagram, circular. But a sickle cell shape, what happens is the circular shape is changed into this moon-like shape. Okay, you can see this like this shape. Okay, the normal is this. This is because of a defect in the gene which makes your RBC, which makes your hemoglobin basically. So now abnormal hemoglobin is made, and because of that, the shape of the RBCs changes, sickle shaped. So that is how the name sickle cell anemia. So when the shape of the RBC changes into this moon-like shape, it can have multiple effects. Okay, it is not normal, it is abnormal. So it can clog the blood vessels, poor circulation of the blood, internal hemorrhages. Okay, internal bleeding can happen, or internal accumulation of blood cells at one place resulting into clotting. That can happen. Okay, so individuals which are heterozygous are normal unless dehydrated or experience mild oxygen deprivation. Okay, we are going to see the cross, so that is where you will understand heterozygous individual. It's like capital T and small t. Okay, hybrid condition having the dominant allele also and recessive allele also. In sickle cell, let's say the normal allele for the circular shaped RBC and the let's say I just represent small n here just for understanding this. So this is a defective gene, okay, which is making sickle cell shaped RBC. This is making normal cell shaped RBC. So here the condition is more or less balanced because one allele is normal, so this can compensate. Okay, so that we are going to see the next slide. Fine. So this is the result of the. These are the major points. What is sickle cell anemia? The shape of the RBC changes because of the defective hemoglobin gene. Okay, this is represented. This is a family tree with respect to the disease which is represented. It is also known as pedigree analysis. So this is the female, circle is represented by female, square is represented by male, and if it is colored or dark shift means it is affected. So the father and the mother is having sickle cell disease, and if she's having these 4 children, 2 daughters and 2. So that is so if this is represented how means what, one gene is normal and another gene is defective. So you are sorry, one allele is normal, another allele is defective. So because one is coming from the half of the chromosome is coming from the mother and half is coming from the father. So this is how the progeny will look like if the mother is having and the children are like this. So heterozygous for sickle cell carrier. Okay. So if half white and half colored, so they are the carrier for the disease. And fully colored, they are the affected. Okay. And if let's say one of the female marries with a male who is also carrier and she is also a carrier, then chances of one of the of getting their progeny or child getting fully affected is there. Okay, because what if this gene and this gene combine, so a female with you know having the disease, that chance is there. Fine. Here the chance was the child having the disease is not there because the male was normal. That is compensated. That is why all the children are carrier. But here, this one of the girl child marries with another male who is also a carrier. So if both are carriers, then one of the child getting the disease is possible. Okay. So let's understand a bit in more detail about the sickle cell anemia. Okay. So here the mechanism, the understanding of the which gene is deleted, so that you should know. On chromosome 11, the hemoglobin synthesizing genes are there. Okay, on chromosome 11. So you have two normal genes, two normal alleles, HBB genes, which will make your hemoglobin. Hemoglobin is the main central iron atom of the RBC, which is a protein pigment which gives red color to the RBC, and it is the pigment which carries oxygen, okay, in the red blood cells and transports it to different parts of the body. Okay, so working beta globulin protein combines with alpha globulin to make hemoglobin. So basically, there are two parts of hemoglobin molecule that is to be made in two parts, alpha and beta. So alpha chain has to be made, alpha protein has to be made, beta protein has to be made. They will come together and they will make something known as hemoglobin pigment. Okay, they will make hemoglobin pigment, and then this is how the normal pigment, normal RBC will look like if both the proteins are made. If both the proteins are made. So alpha protein is also made, beta protein is also made, beta globulin and alpha globulin, they will come together, make the normal circular shape RBCs, hemoglobin filled red blood cells, round and flexible. This is a normal person. Now, what happened if the person is suffering from this genetic disease, sickle cell disease? So, this chromosome 11, which has to produce normal alpha and beta, now it is making alpha chain is properly made. Alpha is normally made, no problem in alpha, but beta is slightly defective. Altered beta proteins, they stick to one another, forming long fibers. Okay, so there is a mutation, a gadbad, a disorder in the in one of the protein which is made, alpha protein. Sorry, beta protein. So these cannot come together properly, and if at all they come together to make the hemoglobin molecule, the RBC will not be of the normal size and shape. It will be some abnormal size and shape like this. So fiber-filled red blood cells become stiff, and they are now this sickle cell shape. This is how the condition that prevails when a person is having a defect in the beta globulin gene, because now it is unable to make the normal beta globulin, beta protein. It is making some abnormal beta globulin protein, and this is how it results in the sickle cell shape. Now we will see the cross what happens when there are parents who are carrier for sickle cell. who are carrier for sickle cell. So this is a parent. This is a gene for the hemoglobin. We know that gene exists two pairs or two alleles. That is, in this case, we will write hemoglobin. So HB represents hemoglobin. Capital A stands for the normal hemoglobin, and S stands for the sickle shaped hemoglobin. So this parent, one, let's say the male and the female, both of them are having carrier. They both are having one allele as defective. Okay, now we'll separate the gametes. So it's very simple. You just write it in a circle form, you separate, and now you will do the cross. So what will happen in F1 progeny? When this allele combines with this allele of this parent, male and female. So HBA and HBA, what are the chances of the progeny? That the child is normal because both the alleles are normal for making hemoglobin molecule, and that is how the RBCs will be normal shape. So this child is not got the disease, nor she is the carrier. Now this A, let's say if this gene combines with this allele of the another parent. So in this case, unaffected, but is the carrier. So the child will not have the disease, but 50% or half the allele, half the partner is defective. So he or she will be the carrier. Another, let's say HBS of this parent combines with HBS of this parent combines with HBA of this parent. So again, unaffected but carrier stage. Half of the allele is normal and another allele is defective. So carrier stage. Okay, some mild symptoms will be there, but that is compensated by this normal allele, which is producing normal RBCs. Mild symptoms will be there, but the disease will not set in. And if HBS and HBS, in this case, the child will have the sickle cell anemia. Will have the sickle cell anemia. So affected because both the alleles are sickle cell gene. And possibility of this child surviving is very, very low. The child might die also. Okay. So again, phenotypic and genotypic ratio will remain the same. 1:2:1 phenotypic ratio. Okay. Unaffected, affected and carrier, unaffected and carrier, unaffected and carrier, and this is affected. 1:2:1, and genotypic is also 1:2:1. These crosses are asked in the exam. You have to know the crosses. You have to practice. Another is, let's do this. Okay, this is the same. HBN and HBS. Sometimes N is also written over here. Okay. So normal, sickle cell trait, and sickle cell anemia. In case of numbers, I have represented over here. So you can be asked different combinations. For example, let's say a sickle cell trait person with a normal with a normal parent. So one can be HBN, HBS, and another can be HBN and HBN. So this cross you can do for your self-study or homework. What will happen if one parent is carrier and another parent is completely normal? So you will do the cross and check it for yourself. Okay. So the F2 generation and F1 generation, F1 generation you will see different combinations. Okay, because here N is there. So let's say if you put N over here, let's just do this over here only. Okay, this is N. Let's say this is we are doing the normal person. One is normal, one is carrier. So in case of this, let's say here I'm putting the N. Okay. So the first will remain the same. Second here, what will happen? This will combine with this, and this N will combine with S. So here, okay, this N and here, N and S here. So in this case, you will see 50% normal and 50% carrier. No child will have sickle cell anemia. So here the phenotypic ratio will be 1:1, and genotypic ratio will also be 1:1. 50% chances of normal, 50% chances of the child being carrier. No affected individual. When one of the parent is completely normal. Here both the examples are talking about when both the parents are carrier. Fine. is all about sickle cell anemia. Now we move to another example, which another topic category where Mendelian genetics is not followed. So another is multiple allelism. Multiple allele means what? Right now, up till now, we know the knowledge is that one gene will have how many alleles? Capital T, small t, or two capital T, or small t. Okay, one gene will have only alternative forms, only two alleles. Okay, one pair, that's it. But there are certain examples, certain scenarios in plants, humans, animals, wherein a gene can have multiple alleles, more than two. More than two, so let's take 3T. Okay, here the example is your blood group. Okay, your example is blood group. So, in which more than two alleles govern the same character. So, same character, same trait, let's say the blood grouping only we are discussing, but more than two alleles can be there. Okay, so the example is ATO blood grouping in humans. So, here how many alleles are there? Not two, three. IA, IB, and I. So if the allele is IA, means the person will have A blood group. If it is B, B blood group. If no A or B, it's just small I, then it will be O blood group. Okay, and by the denotion of this letters, capital, you can make out that these are the dominant ones and small I is for the recessive. Okay, means no antigen, that is A or B, which is represented on the plasma membrane of the RBCs. Fine. So three alleles. Each individual can have only two combinations. Okay. The individual will not have three alleles, but the effect, there are three possible types of allele in this particular scenario. Obviously, the individual will have any two combination, either IA-IB or IA-IA or IB-IB or II or IA into this small IB into small I. Okay, as you can see in this cross. So you can see over here, for example, if both the alleles are with two capital A, then the female is having A blood group. Okay, if the female is if one allele is A and the another is B, then the person will have IB blood group, or AB blood group. If only one capital A and one small I, so A blood group again. So this is the hybrid condition, but one is recessive allele and the another is the dominant allele. Just give me a second. Okay, so each individual will have only two combinations. Number of alleles possible for any gene is constant, but usually more than two alleles exist for any gene in an outbreeding population. So that is what we mean by multiple allelism. Okay, in a population, here we are talking about human blood grouping system. So three combination of alleles are there: IA, IB, and small I. Okay, but in an individual, any of the two combinations can be there. Either IA, I. You can see the combinations over here. Sorry, the laptop cursor is not working. So you can see the cross and you can make it out. Fine. So four different possible blood groups you can see over here. And there's A, AB, B, and O. Okay, depending upon the combination of the alleles. Fine. Another example which is there in your textbook is of the Drosophila. So here also more than one allele is there. All of them are controlling what? All of them are controlling the wings of the Drosophila. Drosophila is a food fly. It is one of the most commonly used organism for carrying out genetic experiments. Yeah. So it is a condition in which more than two alleles govern the same character. So Drosophila, large number of multiple alleles are there. Okay, as you can see the picture. So from normal size to vestigial wings, you can see. Yes, normal wings VG plus. If the Drosophila, the genotype can be VG plus. If the Drosophila is having cut wings, naked wings, then it will be sorry, then VGNI, notched wings. That is, there will be some patterns in between. So VG no. Then strap wings, VGST. No wings, VG. So different ideas you can see of the G. Is having multiple forms: G plus, GNI, GNO, GST. Then nothing on the G. So depending upon what type of G is there, which combines with the V, it will give that particular type of wing to the Drosophila. So this is what this is. The character is the same, that is the wings of the Drosophila, but depending upon what type of allele of G is there with the wing combination, so different types of wings can exist in a Drosophila, from normal to no wings, normal wings to no wings to notch to make to strap. Okay, so that's all for this video. In the next video, we'll see the chromosomal theory of inheritance, the structure of chromosome, and X and Y chromosome. So thank you. Take care. Sorry. We will. There is a problem with my cursor at the moment. So we meet in the next video. Till then, take care. See you. Bye bye.